Welcome to Silent Vibes. In this video tutorial, I am going to discuss about the topic gene cloning. So let's see what is gene cloning, what are the methods and requirements for gene cloning, and what are the steps that are involved in gene cloning. So here we discuss about the topic gene cloning. As the gene cloning means what? It is the replica copy and the replica copy of the gene. Here gene is what? Gene is a basic unit of heredity and a sequence of nucleotides in DNA that encodes the synthesis of a gene product, either RNA or protein. As the gene cloning is also termed as molecular cloning, it refers to the process of isolating a DNA sequence of interest for the purpose of making multiple copies of it. Then the identical copies are called the clones. In 1973, the Stanley Cohen and Herbert Boyer developed the techniques to make the recombinant DNA, that is the form of artificial DNA. Then how the gene is cloned? It is in vivo and in natural process. And the natural process of cloning is what? That is by the replication. And we use this replication in two ways, either in the cell-free or the cell-based DNA cloning. In the cell-based DNA cloning, we use a particular cell that is in vivo machinery, and here the DNA is given to the cell and then it replicates and make a copy. Then the second one is the cell-free DNA cloning. About this, the machinery used is in vitro, where it replicates outside the cell by providing all the circumstances in an artificial environment, and a fragment of DNA containing the gene to be cloned that is inserted into a suitable vector to produce a recombinant DNA molecule. The vector acts as a vehicle that transports the gene into a host cell, usually a bacterium, although other types of living cell can be used. And within the host cell, the vector multiplies, producing numerous identical copies, not only of itself but also of the gene that it carries. And when the host cell divides, the copies of the recombinant DNA molecules are passed to the progeny and further vector replication takes place. Then what are the requirements of cell-based cloning? The DNA fragment is used that containing the desirable genes to be cloned, then the restriction enzyme. Here the selective cleavage of particular gene sequence, and the ligase enzyme is used, that the enzyme that can catalyze the joining of two large molecules by forming a new chemical bond. Then vectors are required to carry, maintain and replicate the gene clone in host cell. Then the host cell in which the recombinant DNA can replicate, and selectable and screenable markers are used. The selectable marker, that is the antibiotic selection, are required to detect the positive cells and are therefore permanently to the host genome, also with a foreign DNA. Then the screenable markers, which are also called as the visual marker, scorable marker or the reporter gene, that will make cells containing the gene look different. And what are the steps that are involved in the gene cloning? The first step is the construction of the recombinant DNA. From the source, we isolate the DNA, then we should digest it by restriction enzyme. It should be endonuclease in nature, as for example EcoR1, and from this we produce the sticky ends. And we have the vector, we should treat it by same restriction enzyme, that is EcoR1. After treating, we get a cut and sticky ends, and then ligate it by the enzyme ligase. And here we obtained the non-recombinant DNA. If the gene of interest is ligated, then we get the recombinant DNA. And hence this is the construction of the recombinant DNA. The second step of the gene cloning is the transformation, that is the natural process. And transformation, it is the process of uptake of DNA by the Escherichia coli. And now we have the tetracycline resistant, which is the non-recombinant DNA. And if the gene of interest is ligated in the tetracycline resistant, then it is the recombinant DNA. Then the transformation of the Escherichia coli means non-recombinant strain and the recombinant strain, which gives the transformed cell. If there is no DNA uptake in the Escherichia cell, it gives the non-recombinant cells. The transformed cells means its foreign DNA enters into the Escherichia coli cells. Either it is recombinant or non-recombinant. The third step is the selection process for transformed cell identification. This is the transformed versus non-transformed cells. So how it is done? It is done either by selectable marker or by antibiotic marker. So the transformed cell having the tetracycline resistant, which is then plating on the tetracycline, then it is survived. And if the tetracycline sensitive is plating on the tetracycline, then it is died. The third step is the selection process for transformed cell identification, that is the transformed versus non-transformed. So how it is done? It is done either by the selectable marker or the antibiotic marker. So here the transformed cell which having tetracycline resistant, that is the non-recombinant strain or the recombinant strain, and if it is plating on the tetracycline, then it is survived. And if the tetracycline sensitive, that is the non-transformed cell, if it is treated with tetracycline, then it is died. So here the selection separates from the transformed and the non-transformed cell. The fourth step is the screening. So the recombinant versus non-recombinant, and this is what? The part of the transformed cell that is either in recombinant or the non-recombinant. To screen this we have the markers, that is GFP, LacZ and luciferase, and these are the screenable marker. And the non-recombinant DNA and the recombinant DNA, by using blue-white screening we get the recombinant strain, and to screen this we use the screenable marker, and hence we screen this by non-recombinant. The fifth step is the amplification. Then what we do? This recombinant strain, then it grows and culture in media. Then the cell duplicates in number from the single cell to double, and 2 cells become 4 and 4 it becomes 8. Then the kinetics, that is n is equal to n naught into 2 is to power n. As per generation we got the increased number of DNA, and this is what the amplification processes does. And so for the gene cloning involves the isolation of DNA, that is the gene of interest, it is fragments and to be cloned, and then the insertion of isolated DNA into a suitable vector to form the recombinant DNA. Then the third step is introduction of recombinant DNA into a suitable organism which is known as host, and then the selection of transformed host cells and identification of the clone containing the gene of interest, and then the multiplication or expression of the introduced gene in the host, then the isolation of multi-gene copies, that is protein expressed by the gene, then the purification of the isolated gene copy or protein, and hence these are the steps of gene cloning. Thank you for watching this video. If you like the video, please do like, share and subscribe our channel, and further vital topics as highlighting CSIR syllabus will be coming, so stay tuned for the upcoming videos.